All right, so previously we have in my previous video, so we have seen pH-pK relationship. So if you have not seen my previous video, which is the very basic one for amino acids, so please watch my previous video. So it is there in the link below. So and continue with this video. So let's get into those 20 standard amino acids, each one of them, and also I will explain you some of the important points related to each one of them. So let's begin now. So our very first amino acid, so let me decrease the font size here. So my very first amino acid is glycine. It has got alpha carbon in the center. It has COO minus primary carboxyl group. It has NH3 plus. It has H, and the side chain of glycine is only H. H is the side chain of glycine, and the glycine you can write it as a three-letter abbreviation as GLY. This is the glycine. As you can note that side chain, that is the R group of glycine, is simply H. That's why glycine is referred as simplest amino acid. That's an importance of glycine. Is the simplest amino acid among all amino acid. Among all amino acid, glycine is the most simplest amino acid, and also H cannot interact with anything. That's why glycine, it is a hydrophobic amino acid. Glycine is hydrophobic amino acid. So, and since it is the simplest amino acid, glycine can be accommodated in a very short space available wherever there is bends, loops, turns, coils. Especially that happens when another amino acid prolene comes in. In that bend, if there is very little space available, usually in that bend, so glycine is accommodated. So along with the prolene, glycine is commonly seen. So that's the importance of glycine. So hydrophobic amino acids are water insoluble. That's why in a globular proteins, hydrophobic amino acids are usually found in the interior core. Or if you are talking about a membrane protein, so the membrane spanning domain, wherever membrane is passing, like a protein is passing through the membrane, in that membrane spanning domain, hydrophobic amino acids will be present. So that's about glycine. So let's put this glycine on the other side. So now let's move on to see our next amino acid, and that is alanine. Now the alanine, it has got alpha carbon in the center, and it has got COO minus, just like any other amino acid, primary carboxyl group. It has NH3 plus, primary amino group. It has H, and the side chain of alanine is CH3. That's a methyl group. That is what, this is the structure of alanine here, and it is simply written as ALA. That's a three-letter abbreviation for alanine. Now note that alanine's side chain is a methyl group, and this methyl group is hydrophobic. So it do not interact with water. So it is a hydrophobic amino acid. So that is the importance of alanine there. So let's put this alanine on the other side now. Now let's move on to see our next amino acid in line, and that is valine. Now let's put the look at the structure of valine. So it has got alpha carbon in the center, as like any other amino acid, and COO minus, primary carboxyl group, NH3 plus, primary amino group. It has H, and it has the side chain, and the side chain of valine, very interesting. So it has a branch point here. So CH3 methyl group and another CH3 methyl group. So that means, so the side chain of valine has a branching here. So there is a branching. That is what is the feature of valine. So valine, it has a three-letter abbreviation VAL. Okay, so and since it has methyl groups in the side chain, so valine is hydrophobic. So valine is one of the hydrophobic amino acid. And also valine has a branch here. That is why it is grouped under branched chain amino acids. So valine is one of the branched chain amino acids. So branched chain amino acid is valine. Okay, and also note that all branched chain amino acids are consumed by skeletal muscle proteins are the major consumers of branched chain amino acids. So the kind of proteins that we see in skeletal muscle, so they are containing predominantly branched chain amino acids. So that's why skeletal muscles consumes more branched chain amino acid than other tissues. So that's about valine. So valine is a hydrophobic amino acid. Okay, now let's move on to see our next amino acid, and that is leucine. So again, leucine, like any other amino acid, so it has got alpha carbon in the center, it has COO minus, it has NH3 plus, like any other amino acid, it has H. And now look at the side chain of leucine. So we are talking about leucine now. We have done with the valine. Leucine, it has CH2, and then you see the branch CH3 and CH3. So there is a branching like valine. Leucine also has got a branch. Only thing is one of the CH2 is more than valine. So this is what is leucine. Leucine is again a branched chain amino acid, and 3-letter abbreviation for leucine is LEU. And it is a branched chain and hydrophobic, just like valine. So it is also hydrophobic amino acid, and it belongs to branched chain category, just like valine. And it applies to, like the point that I told, skeletal muscle proteins contains more of branched chain amino acids. Now let's move on to see our next amino acid, and that is isoleucine. So isoleucine, let me move this somewhere. Yeah. So isoleucine, like any other amino acid, alpha carbon in the center, and then it has COO minus, primary carboxyl group, primary amino group, H is there. And now isoleucine, it has got carbon with the CH3 attached and H, and then there is a branch again, CH2 and then CH3. So there is a branch point in isoleucine. How the branch point? As you can see, if you carefully look at this particular side chain R group, so there is a branch here. So this side, and then the branch is down here. So this is what is isoleucine. And the isoleucine, like valine and leucine, it is also hydrophobic amino acid. So isoleucine is hydrophobic because these methyl groups do not interact with water. It is hydrophobic amino acid, and also it is the branched chain amino acid. So that means skeletal muscle proteins are the major consumers of isoleucine, valine and leucine. So these are 3 branched chain amino acids. Now let's move on to see our next amino acid, and the next amino acid that we have here is sulfur-containing amino acid. So sulfur-containing amino acid, so we have 2 of them. So one is methionine. Let's look at methionine first. So methionine, like any other amino acid, it has alpha carbon in the center, COO minus, primary carboxyl group, NH3 plus, primary amino group, H. And now look at the side chain of methionine. It has CH2, and then there is a sulfur. It has got sulfur, and then again so CH3. This is the side chain of methionine. So the methionine side chain is CH2, sulfur and CH3. So it is a sulfur containing amino acid. So methionine is sulfur containing amino acid. And also note that methionine sulfur, it is not free to interact with any other amino acid or any other molecule. So sulfur is not free, and also it doesn't interact with the water. So that's why it is a hydrophobic amino acid. So methionine is an hydrophobic amino acid. What is the importance of methionine in our body? So the methionine, eventually it will be in a metabolic process. You are going to study that in biochemistry. So the methionine, it will be converted to what is called as S-adenosyl methionine. S-adenosyl methionine, sometimes we just write it as SAM, S-A-M, S-adenosyl methionine. What is the importance of SAM? So the SAM, it participates in methylation reaction. in methylation. So we have methylation reactions in our bodies. One of the example for methylation reaction is DNA methylation. DNA methylation is the methylation means the methyl group that is attached in DNA methylation process. Usually that will come from S-adenosylmethionine, which is a derivative of amino acid methionine. So the methionine is the one that is going to make S-adenosylmethionine, which is involved in methylation reactions. That's the importance of methionine, one of the sulfur containing amino acids. Let's move this methionine on the other side. Now let's move on to see our next sulfur containing amino acid, and that is cysteine. So the cysteine, like other amino acids, so it has alpha carbon in the center, it has COO minus, NH3 plus, it has H, and then it has a sulfur. Now as you can see, it has CH2 in the side chain and it has SH. This is the side chain of cysteine. So the cysteine, it is a sulfur containing amino acid. Because it has sulfur, cysteine is sulfur containing amino acid, and sulfur is free here. It can interact with another cysteine molecule. Let's see how that happens. So let's move this cysteine down here. And also note that cysteine can be written as CYS cysteine, like methionine written as MET methionine. Cysteine is CYS. Anyway, so how the disulfide bridge is created here between two cysteine molecule, let me explain that to you. So one of the cysteine, so we have alpha carbon of cysteine. So let me write NH3 this side now, NH3 plus, COO minus, H, and then CH2, SH, that is our cysteine, one of the cysteine. So another cysteine, I'll write here. So CH, NH3 plus, COO minus, then we have CH2 and SH. Okay, now if you, this is another cysteine molecule in the blood. These two cysteine molecules, they can interact with one another and make a disulfide bridge. So what happens is we have COO minus, H, NH3 plus, CH2, S, and from this side another S, CH2, carbon, alpha carbon, NH3 plus, COO minus, and H. So basically what we are seeing here is the two cysteine molecules are joined with one another by a disulfide bridge or the disulfide linkage. So the disulfide linkage is the one that brings two cysteines together, and this entire molecule, which is basically oxidation of two cysteine molecules, is now we call that molecule as cysteine. So basically cysteine is an oxidized form of two cysteines. Now the importance of this is imagine in a polypeptide chain, if you have two cysteine molecules a little bit apart in a polypeptide chain, so they can combine with one another and create a disulfide bridges. So let me write that to you so that you understand it better. So let me delete this part here, so erase it so that I have some space here. So how that this happens. So here is your let's write, say, consider this is the polypeptide chain, and one of the amino acid here is consider your cysteine, CH2SH, that's one of the cysteine. Another cysteine is down here, CH2SH. Now these two cysteine molecules, they can interact with one another. So basically you take these two H out. If you remove that, so what is remaining is if you take this H out, so they both can combine, they make a disulfide linkage, and that's how they both connect with one another. Because of this, what happens? What will happen to the polypeptide chain? So polypeptide chain, it comes closer, right? So CH2S double like disulfide linkage CH2S. So basically it helps in conformational change in a polypeptide chain, and thereby it helps in protein folding process, having a tertiary structure or something like that. That's how the cysteine formation, oxidation of two cysteines will help to make a disulfide linkage. Now let's move on to see our next amino acid. It is one of the important one called Prolene. Now the Prolene has got alpha carbon in the center. It has COO minus, that is a primary carboxyl group, and then it has the side chain which is special here. So it has NH plus instead of NH3 plus, because the side chain of Prolene, it will interact with the another Prolene molecule. So the side chain of Prolene is going to interact with the prime amino group present in a cysteine molecule. This is how it happens. So CH2 and it make a linkage like this. So basically this amino group is going to interact with the side chain of Prolene. See, the Prolene side chain is CH2. It is going to interact with the amino group that is attached to the alpha carbon. Normally, as you have seen in other amino acids, so alpha carbon amino group, alpha carbon carboxyl group, they both are free. Whereas in Prolene, carboxyl group is free, that is a primary carboxyl group. Whereas in Prolene, amino group is not free. It is attached to the R group. It is attached to the side chain. That's why this amino group, we don't refer it as a primary amino group. Rather, we refer it as a secondary amino group. So the amino group that is present in Prolene is referred as secondary amino group, just to differentiate from other amino acids, because this is special because it has interacted with the R groups and made a 5-membered rigid ring. So it has a rigid ring here by attaching to the R group. And also because of this rigid ring, so when the polypeptide chain is made, so whenever Prolene comes in, so the rigid ring is not accommodated properly in a trans configuration of a polypeptide chain. Rather, it makes a cis configuration, thereby the polypeptide chain, whenever Prolene comes in, it is going to change the direction of a polypeptide chain. That is why Prolene is sometimes we refer it as helix breaker, because it do not allow alpha helix to continue. That's why it is called as helix breaker. It leads to, Prolene leads to bends, loops, turns and coils. Bends, loops, turns, coils in a polypeptide chain is all because of Prolene. That's the importance of Prolene. So because it is such a different amino acid compared to all other amino acids, that's why Prolene sometimes it is referred as amino acid rather than amino acid. Prolene is sometimes referred as amino acid. So these are some of the points related to Prolene. Now let me go back to the previous slide. So also Prolene is it's a hydrophobic amino acid. Prolene is hydrophobic. So it do not interact with water. So this is the hydrophobic amino acid. Now let's see, and also let's move on to see our next group of amino acids, and those are phenylalanine, tyrosine, and tryptophan. Let's see all of them together. Now the phenylalanine, so just like any other amino acid, so it has alpha carbon in the center, it has COO minus, it has amino group NH3 plus, it has H, and now the R group in phenylalanine is CH2, it has CH2, and then it has a benzene ring. That's the phenylalanine. So the side chain of phenylalanine is CH2 and benzene ring. Okay, it's an aromatic ring. So aromatic, benzene ring is sometimes it is referred as aromatic ring. So now let's move on to see our next amino acid in the same category. So it is tyrosine. So it has alpha carbon, COO minus, primary carboxyl group, NH3 plus, primary amino group, H, and then CH2, just like phenylalanine. This is phenylalanine, and the 3-letter abbreviation for phenylalanine is PHE, phenylalanine. Now tyrosine, it has CH2 benzene ring, just like phenylalanine, it has a benzene ring. Additionally, it has hydroxyl group attached to the benzene ring. That is what is tyrosine is. Tyrosine, and it is written as TYR, 3-letter abbreviation. So the side chain of tyrosine is CH2 benzene ring and hydroxyl group. Now our next amino acid in the category is tryptophan, alpha carbon. It has COOH, COO minus, NH3 plus, and then CH2, and then it has got a 5-membered ring with the nitrogen attached, and then further this will be attached with benzene ring. So this is what is tryptophan here. So tryptophan side group is, it has CH2, 5-membered ring and the benzene ring. This is tryptophan. And tryptophan, you can write it as TRP, 3-letter abbreviation. Now what is common with all these 3 amino acids? So they all have aromatic rings. That's why these are referred as aromatic amino acids. So we have 3 aromatic amino acids, and those are phenylalanine, tyrosine and tryptophan. Now come to some of the important points here. Now the phenylalanine, it has got hydrophobic benzene ring, so it is hydrophobic amino acid. Phenylalanine is hydrophobic. Tyrosine, since it has got hydroxyl group, so it is a hydrophilic amino acid, so it doesn't belong to hydrophobic. And also note that tyrosine, since it has hydroxyl group, so it can participate in phosphorylation. One of the post-translational modification is phosphorylation. This phosphorylation, sometimes it can go on over the hydroxyl group in the protein molecule. Now the tryptophan, it has largely it is hydrophobic compared to hydrophilic capacity. Hydrophobic. That's why I will put this tryptophan into hydrophobic amino acid category. So of course, there in the metabolism of phenylalanine, tyrosine, tryptophan, so there will be certain disorders. Anyway, we are going to go into that later as we move on with other chapters in biochemistry. So these 3 are aromatic amino acids. So out of that, phenylalanine and tryptophan are all hydrophobic amino acids. Okay, so that's about aromatic amino acids. Now let's move on to see our next category, and that is we are going to look into other hydroxyl containing amino acids. Now other than tyrosine, so we have other hydroxyl containing amino acid is serine. So serine has COO minus, it has NH3 plus, H, and then in the side chain serine has got CH2OH. This is the side chain of serine, CH2OH. As you can see, there is a hydroxyl group. So that is what is the serine. Serine is a hydroxyl containing amino acid, so you can write it as SER, serine. Now one of the post translational modification is the glycosylation or the glycation, especially O-linked glycation on a protein molecule. It is a glycoprotein or proteoglycan. O-linked glycation, it is generally happens here on the hydroxyl group of serine. And then similar to serine, we have another hydroxyl containing amino acid, and that is threonine. Now the threonine has got alpha carbon in the center. It has COO minus, it has NH3 plus, it has H, and the side chain of threonine, it has got COH hydroxyl group, and then it has the methyl group here, CH3. So this is what is threonine. So threonine is one of the hydroxyl containing amino acid, and 3-letter abbreviation for this is THR, threonine. So threonine, hydroxyl containing amino acid, so it will participate in post translation modification. So that's the importance of threonine. So we have 3 hydroxyl containing amino acids so far, that is tyrosine, serine, which is involved in O-linked glycation, and threonine. These are the 3 hydroxyl containing amino acids. Now let's move on to see our next amino acid category, and that is acidic amino acids. Now we have 2 acidic amino acids. One of the acidic amino acid is aspartate. Let's write the structure of aspartate. COO minus, primary carboxyl group, primary amino group H, and then the side chain of aspartate, it is CH2 and COO minus, that is the carboxyl group. Now see the change here. So the R group, that is the CH2COO minus, it has a negative charge here, and also there is a negative charge on the primary carboxyl group compared to the positive charge that is present on the amino groups. That means aspartate has 2 negative charge and 1 positive charge, so that means it has a net negative charge under physiological pH. That's why aspartate is referred as acidic amino acid. Aspartate is an acidic amino acid, or it is a negatively charged amino acid. So R group, basically the pK of aspartate is less than the the pH is less than the pK here, so that's why there is a deprotonation. So the pH is more than the pK here. R group pK is less, that's why it has underwent a deprotonation. So thereby it has got a net negative charge. So that's about aspartate. It's a negatively charged amino acid. We have another similarly negatively charged amino acid, and that is glutamate. Let's look at glutamate. It has got alpha carbon in the center, COO minus, NH3 plus, and H, like any other amino acids. Then you have CH2 and COO minus. This is what is glutamate. Now the side chain of glutamate is CH2 COO minus. That means it has got a negative charge in the R group. R group has a negative charge, and we have one more negative charge on the primary carboxyl group. So 2 negative and 1 positive charge on on glutamate. So it has got a net negative charge. That's why glutamate is also acidic amino acid, or a negatively charged amino acid. So that's about 2 negatively charged amino acids. Now let's move on to see our next category. So our next category of amino acids are positively charged amino acids. Let's look at positively charged or basic amino acids. So our first basic amino acid is the arginine. So we have arginine has got alpha carbon in the center, COO minus, NH3 plus, H, and then the side chain of arginine is, it has CH2 to 3 times, and then you have NH, and then C carbon with the double bond NH2 plus, and then it has NH2. This is the side chain of arginine. Our amino acid here is arginine. Now look at the charge of arginine. So it has 1 positive charge in the primary amino group, another positive charge in the R group. So R group has a positive charge there, and 1 negative charge. So 2 positive and 1 negative. So that means it is a positively charged amino acid, which is a basic amino acid. Arginine is a basic amino acid. Now look at the one of the special part of the group here in arginine. This part, NH, C double bond NH2 plus and NH2. This special part, we call this as a guanidinium group. Guanidinium. Arginine has guanidinium group. So the guanidinium group is the one that will provide a positive charge to an arginine amino acid. That's about arginine. Now let's move on to see our next basic amino acid is lysine. The lysine structure, again it has COO minus, primary carboxyl group, primary amino group H. Now look at the side chain of lysine. It has 4 times CH2. So CH2, 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 and then it has NH3 plus amino group. So this is what is lysine. So the lysine, it has 2 positive and 1 negative charge. So the side chain of lysine is this. Since the side chain or R group of lysine has got an additional positive charge, that's why lysine is a basic amino acid. So now note that both arginine and lysine, they are strongly basic amino acids, strongly basic. So they are always positive under physiological pH. So both lysine and arginine are strongly basic amino acid. Now we have another basic amino acid called histidine. Now histidine is something that is special here. So the alpha carbon COO minus NH3 plus H, and the side chain of histidine, it has CH2, and then it has a 5-membered ring. It is C double bond C here, and then nitrogen with the hydrogen attached, CH, and then nitrogen which is making a bond with the carbon here. So this is what is the side chain of histidine. So side chain of histidine is this one, and this side chain we refer it as imidazole group, imidazole. So the histidine, this entire thing is histidine, amino acid histidine. So histidine has got imidazole group. Now as you can see, there is no positive charge mentioned here. Why this histidine is, now we are saying it as a basic amino acid. So because we are studying histidine into a basic amino acid category, basic amino acid, but you don't see any positive charge there. So no additional positive charge. That's an importance of histidine here. So in order to understand that, we need to know what is the Pka of histidine. So the Pka of histidine, when it is part of a polypeptide chain, it is 7.0. Now the physiological pH, as we know, so the pH of blood, physiological pH is 7.4, and the Pka that we are referring here, it is 7.0. So 7.0 and 7.4. So if you look at that, so pH is almost equals to Pka, almost 7.0 and 7.0. So when the pH is almost equal to Pka, so that means 50% of this R group of histidine is protonated, 50% of them, and rest 50% will be deprotonated. The molecule that you are seeing here is deprotonated. There is no proton attached. So that's why you don't see any positive charge. So that means we are looking at this 50% of histidine. Rest 50% of the histidines, there will be NH plus there. So there will be attachment of H here, NH plus. So that's how it will be. So that's protonated, 50%. So 50% of histidine molecules will be protonated, 50% will be deprotonated under physiological pH 7.4. That's why, so since the histidine retains protonation and deprotonation capacity, so that is why histidine can act as a buffer. So buffers are basically, they resist change in the pH. So the presence of histidine in a polypeptide chain, it will contribute to buffering capacity of protein, buffering of proteins. That buffering capacity of proteins, it will come from histidine, because histidine has physiological R group Pka of histidine is 7.0, which is closer to 7.4. That's why it has got a physiological buffering capacity. So these are all the amino acids that we have seen. So we have 20 standard amino acids. So in these 20 standard amino acids, so I have explained you some of the importance of all these amino acids. Now let's look at classification. So just to go over again, so we can classify amino acids as hydrophobic, one of the category, hydrophobic, amino acids. Hydrophobic amino acids you can remember as pimp gal TV. Pimp gal TV. So pimp gal TV, so it says basically the P for phenylalanine, I is for isoleucine, M for methionine, another P for Prolene, G for Glycine, A for Alanine, L for Leucine, T for Tryptophan, and V for Valine. So this is what is pimp gal TV, hydrophobic amino acids: phenylalanine, isoleucine, methionine, Prolene, Glycine, Alanine, Leucine, Tryptophan, and Valine. All these are hydrophobic amino acids. And in a globular protein like protein which has globular shape, all hydrophobic amino acids will be arranged in the interior core of a globular protein. They are present in the core. If you are referring to a protein which is part of cell membrane, so as you know, cell membrane is a lipid membrane, and if you are talking about a protein which is passing through the membrane like a transporter, so the protein that is part of a membrane, so in this part, membrane spanning portion, like the protein that is spanning the membrane, so this transporter which is passing through the membrane, so in this particular portion there will be hydrophobic amino acids. Hydrophobic amino acids are present in this middle portion there, in the membrane spanning domain of a transmembrane protein. On either side there will be hydrophilic amino acids. That's one of the classification of amino acids. So our next classification of amino acid is sulfur containing amino acid. Sulfur containing amino acid, you know, it is methionine and cysteine. These are the two sulfur containing amino acid. We have OH containing amino acid, hydroxyl containing amino acids. Hydroxyl containing amino acids are serine, threonine, and tyrosine. These are the three hydroxyl containing amino acids. Aromatic amino acids. Aromatic amino acids, as you know, it is tyrosine, phenylalanine, and tryptophan. Tyrosine, phenylalanine, and tryptophan. So acidic amino acids. Acidic amino acid is, we have aspartate and glutamate. These are the two acidic amino acids. Basic amino acids can be classified into strongly basic, that is arginine and lysine are strongly basic, and the weakly basic amino acid is histidine. Okay, so these are some of the other categorization of amino acid. And one of the the two amino acids that I forgot to explain while I was explaining amino acids, and that is asparagine and glutamine. So let me write down the structure of asparagine. It has COO minus, NH3 plus, H, then it has CH2, and then C double bond O and NH2. So this is the side chain of asparagine. And asparagine, it will be written as ASN. Asparagine. The importance of asparagine is in a polypeptide chain. So imagine that it is part of a polypeptide chain, and the side chain, that is especially the amino group of asparagine, it will be attached with the, say, glucose. So I'm going to take this out and add glucose to this side chain. So if the glucose is attached to the side chain, say carbohydrate glucose is attached, another glucose attached, and then the galactose attached, this part is called as glycosylation or glycation or glycosylation, which is attached to the nitrogen of asparagine. This type of glycosylation we call it as N-linked glycosylation. So usually this happens in the lumen of the endoplasmic reticulum. In the rough endoplasmic reticulum lumen, this glycosylation will happen, N-linked glycosylation. it involves asparagine residue. Okay, so that's about asparagine. Let's move on to see our last amino acid, which I forgot to explain you earlier, and that is glutamine. So glutamine has got alpha carbon in the center, COO minus, NH3 plus, H, and then it has CH2, and then C double bond O and NH2. That's the amino group here. That's the side chain of glutamine. Glutamine, you can write it as GLN, glutamine. Now what is the importance of glutamine? Now the glutamine, since it has got this amino group in the side chain, this amino group can be donated to make amino sugars. So amino group, if it will be given to say glucose, so that will become glucosamine 6-phosphate, and this glucosamine 6-phosphate, it will be converting into different types of amino sugars. So basically the sugar, the glucose, it will take amino group donated by glutamine. Glutamine is the one that donates amino group to carbohydrate to produce different types of amino sugars. Amino sugars are part of glycoproteins and proteoglycans. So all amino sugars that you see in glycoproteins and proteoglycans, the amino group of all those amino sugars will be derived from glutamine. Glutamine is the donor of amino group present in amino sugar. Remember that. So this is all about some of the important points related to each of the amino acids, 20 standard amino acids that we have seen so far. So this is one of the classification of amino acids based on the side chain or the structure. Based on the side chain or the structure of amino acids, we have classified amino acids. So in my next video, I will be explaining you other classification of amino acid, and that is classification based on the nutritional requirement of amino acids. So the nutritional classification of amino acids, I'm going to explain you in my next video. So please watch my next video, so that that will be the end of amino acid lecture. So it is there in 3 parts: amino acids, pH-pK relation, classification of amino acid based on the structure, and the next is classification of amino acid based on the nutritional requirement. I will see you in my next video.